Hi to all, it is a great pleasure for me to have the opportunity to present the work of my master degree dissertation, a project that I developed in the last 2 years in the laboratory of Professor Protasi at the University of Chieti. We will talk about heat strokes, which are crises characterized by an abnormal increase in core body temperature above 40 to 41 degrees, triggered by high environmental temperature combined or not with the humidity. And one of the factors that can predispose individuals to hyperthermic crisis is also intense physical exercise carried out in adverse climatic conditions. And in humans, there is another syndrome characterized by abnormal increase in body temperature called susceptibility to malignant hyperthermia, a rare disease triggered by halogenated anesthetics such as halothane, commonly used in emergency room. And most but not all human families affected by MH have been linked to mutation in gene encoding for ryanodine receptor type 1, a protein that encodes a tetramer forming the calcium release channels in sarcoplasmic reticulum of skeletal muscle. And for more than 10 years, the laboratory of Professor Protasi has been working, thanks to funding from Italian Teleton Onlus, on a project that aimed to demonstrate that hyperthermia triggered by anesthesia and crisis induced by environmental heat and physical exercise are the same skeletal myopathy, just triggered by different stressors. And a correlation between malignant hyperthermia triggered by anesthetics and environmental or exceptional heat strokes has been hypothesized in literature for a few decades as cases of exercise-induced rhabdomyolysis and sudden death have been previously reported in individuals with a family history of MH. It is now generally accepted that hyperthermic crises are the result of unbalanced calcium handling in skeletal muscle fibers, which is controlled by different mechanisms. And first, excitation contraction coupling, a mechanism that controls the release of calcium from ryanodine receptor, which is the protein mutated in family linked to MH. And this mechanism occurs in specialized intracellular junction formed by the association of sarcoplasmic reticulum, a system of intracellular membrane that accumulate calcium, and the transverse tubule, which in the figure you see as an invagination of the external membrane, which contain the DHPR voltage sensor. And the calcium released during easy coupling activates muscle contraction. That then is quickly removed by Cerca pumps and also in part by plasma membrane ATPases to allow relaxation of fibers. During this cycle, small amount of calcium enters mitochondria to stimulate aerobic ATP production. And but when the muscle are stimulated for a long time, for example, during a session of prolonged exercise, DSR may undergo depletion, a condition that contributes to fatigue. And when SR calcium levels are too low, fibers have mechanism to recover the calcium lost from the extracellular space, and one of them being store-operated calcium entry, which you heard about yesterday in the presentation of Professor Protasi, my supervisor. SOCI is calcium influx pathway found in most excitable and unexcitable cells, which is activated by the depletion of intracellular stores, and is mediated essentially by two proteins. STIM1, which is a calcium sensor placed in the membrane of endoplasmic or sarcoplasmic reticulum, and ORI1, a calcium permeable channel of the external membrane, which in muscle is placed in the transverse tubules. And SOCI in muscle plays an important role to limiting fatigue during repetitive stimulation, and has been also proposed to be important during muscle development and growth. But despite the importance of SOCI in skeletal muscle physiology was never argued, the intracellular sites of interaction between STIM1 and ORI1 has been long debated. And initially, SOCI was proposed to occur in the triads, the structure also responsible for the release of calcium during easy coupling. However, findings collected by our laboratory in the last 5 years that leads to 3 independent publications between 2017 and 2020 indicates that SOCI occurs in junction that assemble during exercise that that increases the colocalization between STIM1 and ORI1 and that enhance the recovery of divalent cations from the extracellular space during repetitive stimulation. And this junction modeled in this cartoon, in which STIM1 and ORI1 are shown respectively in red and green, has been named by our laboratory calcium entry units. And these 3 EM images show that they are placed next to triad at the I band, small in control fiber, as you see in this panel A, but get larger and more frequent following acute exercise on treadmill or long-term training in wheel cages. And so from the discovery of calcium entry units, from what we know about the molecular basis of the exertional heat strokes, we developed the hypothesis that drove the experiment for my master's degree dissertation. And the question was, the assembly of calcium entry units could be a factor that increases the risk of heat strokes when exercise is performed in challenging environmental conditions. And for this project, we used adult wild-type mice that were divided in 3 different experimental groups. We have the control mice. The mice trained for 1 month by voluntary running in wheel cages, and mice just pre-exercise 1 hour with a protocol that we already know promote the assembly of calcium entry units. That then these 3 groups of mice were subjected to an extracellular stress protocol in vivo. So a treadmill was placed in a climatic chamber where the temperature and the humidity were kept respectively at 34 degrees Celsius and 40%. And then the mice were subjected to an incremental running protocol where the speed was increased from 5 meters per minute, and this is the speed you see in this video, up to 30 meters per minute. And during and after the exertional stress protocol, we performed the experiment that you see listed here. And the first data we collected was the rise in internal temperature of animals. So we measured the core temperature of animals immediately before and after the exertional stress protocol. And in panel A, you see the temperature at the end of the exertional stress protocol. And in panel B, what is shown is the difference between the initial and the final temperature recorded. And this result showed how the 2 groups of animal trained for 1 month and exercised for 1 hour reached a higher final temperature compared to that of control. And this difference was significantly, statistically significant. We then used a widely recognized test for the assessment to the susceptibility to malignant hyperthermia, such as the in-vitro contracture test or IVCT. And so the EDL muscle were dissected from the animals and subjected to an ex vivo exertional stress protocol consisting of 150 consecutive tetani, each lasting half a second at 80 Hertz applied every 10 seconds. And the experiments were performed in a solution containing 2.5 millimolar of extracellular calcium, but supplemented with BTP2, a SOCI potent inhibitor, in those traces shown in red. And during the exertional stress protocol, the EDL dissected from trained 1 month and exercise 1 hour mice developed a relative basal tension indicative of a contracture greater compared to that of control. And this contracture was significantly reduced by BTP2 in all 3 groups of samples. And the drop in the development of tension due to the supplementation of BTP2 gives a direct measure of the contribution of extracellular calcium to the development of a contracture. So, as calcium entry is mediated by calcium entry units, junction that assemble during exercise thanks to the remodeling of the SR, which form the SR stacks shown by these empty arrows and the inset in panel B, and thanks to the elongation of the T-tubule at the I band shown by the black arrows and the inset in panel C, we performed quantitative analysis by electron microscopy of these 2 structural elements, and analysis of the length of contact between T-tubules and the SR stacks, which is indicative of a fully assembled calcium entry units. So we analyzed EDL muscle before and after the exertional stress protocol. And the EDL from train 1 month and exercise 1 hour mice contain a higher percentage of fiber with the SR stacks and the number of SR stacks per cross-sectional area compared to that of control, already in basal condition. And both parameters increased following the exertional stress protocol in all 3 groups of samples, though remaining lower in control specimens. We then evaluated the T-tubule elongation at the I band and its contact with the SR stacks. So the results shown in these panels indicate that the T-tubule elongation at the I band and its contact with the SR are greater in muscle from train 1 month and exercise 1 hour mice compared to that of control, already in basal condition, but also following the exertional stress protocol. And noticeable the fact that the T-tubule elongation at the I band is greater in a sample from mice just pre-exercise 1 hour before the exertional stress protocol. And this is likely because of a fast plasticity of the T-tubule and a boost effect of 2 close incremental running protocol to which these mice were subjected. In this final slide, we summarize the main result obtained in the study, and altogether our data suggests that the assembly of calcium entry units following exercise or long-term training, which are structures that promote the entry of the recovery of calcium entry of calcium from the extracellular space, which is shown in this graph as a rise in relative basal tension of EDL muscle, predisposed to an abnormal increase in core body temperature when exercise is performed in challenging environmental conditions. And I would like to thank the members of the lab who helped me in different aspects with in this project, the founding agencies, Teleton and NIH, and all of you for being online today. Thank you.
Okay, thank you very much. Excellent talk that covers many interesting conditions because you covered exercise, malignant hyperthermia, and this heat stroke that is, sometimes you can see this rhabdomyolysis in patients. Let me just ask, what are these stacks that you see, you know? Because I'm kind of confused when you talk about all this movement of the T-tubule, because sometimes you talk about proliferation of the sarcoplasmic reticulum. Sometimes, obviously, we have the T-tubular aggregate. What are the stacks? Hypertrophy of the sarcoplasmic reticulum, how do you define them?
We found that sarcoplasmic reticulum, after a depletion in his stores, reassembly is structured in this flat cistern of DSR And the T-tubule elongate from the triads to into this structure to promote the recovery of extracellular calcium in this in the muscle.
Yeah, but they can interfere with calcium. But at the end, you have in hyperthermia and in stroke, in heat stroke, you have production of heat. So you must have some kind of calcium exit, a massive uncoupling of the mitochondria. What is your connection between these three conditions?
That's kind of a, I don't know, Barbara, if you want me to say something.
Yes, please.
We, Simona and our laboratory together with Antonio Michelucci, we found that following exercise, we have the assembly of these units that we think mediate calcium entry. And this unit also disassemble following exercise. In the following 24 hours, they disassemble, at least when we exercise acutely for 1 hour. We have several lines of evidence that indicates that the assembly of these units allows calcium entry and reduces muscle fatigue. Meaning that when these junctions are assembled, the fatigue can be slightly less and delayed, okay? This assembly of the units doesn't work very well in aged muscle, actually, because in aged muscle, these units are absent. And indeed, aged muscle, as Simona showed this morning, is unable to use this external calcium. Tubular aggregates are different from this. Tubular aggregates are found in myopathies or are found in aging, but they also accumulate a lot of STIM1 and ORI1. And when we have tubular aggregates, socket doesn't work. So now the talk of Barbara is saying that the assembly of these units when we do exercise, if we exercise a lot in heat, they can actually increase a lot the calcium entry, and that this can put at risk the subject to heat stroke. Indeed, when there is a marathon, the weather is very hot,
yeah,
a lot of people are ending up in the emergency room.
Right.
So this the assembly of calcium entry unit is positive for muscle. However, when we exercise in the heat, maybe is an additional factor of risk. Animals usually, when it's very hot, they don't do what humans do. You know, they hide. But humans sometimes they do exercise even when it's very hot, and maybe it's not such a great idea. So.
I prefer hot water, but you didn't answer the question of Professor Angelini. What is the meaning of stacks? You never used the stacks.
Oh, stacks. New. Flat cistern, more than one. We call them stacks. In the old times, stacks were already described in literature, and they were described as SR stacks, but nobody knew that there was a transverse tubule between them. When we exercise, we have sometimes SR stacks may exist even without the transverse tubule. And the stacks, we think that they have a lot of steam.
And the stacks are the different layers of sarcoplasmic reticulum?
SR.
SR.
Yes, sarcoplasmic reticulum.
Cistern of SR.
Yeah.
Yeah.
I have a question. May I?
Yes.
So you didn't mention about the membrane disruption at all. So what do you think about membrane disruption? So according to our data, so if we gave exercise, usually it's downhill exercise. So bicep usually, so maybe 5 to 30% myofibers were injured. So what do you think about that? Can you explain about that?
Do you want to say something, Barbara, or should I go? I mean, you mean during the exertional stress, do we have some damage?
Yes.
That's a yes. Obviously, we have some damage. We are quantitating the damage. I don't think that Barbara has finished the count.
No, we have not. No, we have not finished to calculate the damage.
So
fibers.
How can you calculate the damage? Membrane disruption and so advanced blue, or so how can you?
How do we calculate damage?
with the alteration of the structure in the the orientation of the sarcomere in the fibers. So the percentage of the fiber altered and not altered.
So we measure using histological section and the M. Simona, do you want to comment on this? How do you measure the damage?
Well, if I recall, we did something in another paper, the exceptional stress.
Yes.
With Cecilia and Paolini and Michelucci. So when the myofibrils are completely disrupted, right?
Mm-hmm.
And but I don't think we have, yeah, I don't think we have looked at specifically at the membranes at that time. We did. I don't remember though.
No.
Okay.
So what we look is loss of striation and
yeah,
damage.
That's it. Yeah, but not at the membranes, right?
Yep.
Okay.
So we are interested in damage and repair. So you know, so not just the disruption only. So we are interested in receiving plasma membrane. So that's why.
I've seen your talk. I enjoyed your talk a lot.
Yeah. Thank you.
So no, we never look specifically at the plasma membrane or sarcoplasmic reticulum membrane specifically.
But we have indications though. In our studies, usually what we do is measurement in the blood of the level of CKs, calcium, potassium. So we
yes,
muscle damage,
that is higher.
Blood parameters of CK.
Yeah, in all this condition, you have a massive variation of CK.
Yeah.
And sometimes in malignant hyperthermia, anesthesiologists like to look at CK, but
yeah, that's what we do in mice. When our mice have a hyperthermic crisis, we look at the level of CKs. We have published this several times.
Yeah.
So in the case of Barbara experiments, we see an elevation in temperature, but we don't have the heat stroke yet. There is no heat stroke there, just a more increased internal temperature in those mice that have these junctions assembled, indication, and more tension. Indication that, you know, these calcium entry units can underline a greater increase in temperature of the muscle.
One more question. So how about cold condition? Because membrane cannot reseal at the cold temperature. What do you think about cold?
Cold.
Oh, what do you mean about cold?
Yeah, cool. You know, so
lower temperature.
Lower temperature.
Lower temperature.
Anything with lower temperature. By a few years ago, when I was in Boston and I worked in Paul Allen's lab, we did the experiments of rapid cooling.
What happened?
By expressing RNOD receptor 1, 2, and 3. And actually all three isoforms of the RNOD receptor with rapid cooling release a great amount of calcium. That's the only thing I know.
So cooling is also bad.
It's also bad.
Experiment was done in vitro and it was very low temperature. I mean, we really were putting in a solution very cold, but the release of calcium was massive.
Yeah, but they don't, I mean, it's a reactive form, but we don't know if it is effective.
Yeah.
Well, there's a lot to understand about hot, cold, malignant hyperthermia, all these formations, CEU, CRU. Congratulations, Feliciano, for this nice work. Any other question?